Okay, so in the last segment, we went over what linkage is, what it means, and that you would stumble across it when you're doing a dihybrid cross, but you do not get a 9 to 3 to 3 to 1 ratio in the offspring. So if that happened, then you would back up a little bit and do a different kind of cross to help you figure out what the distance is. We call it the genetic distance between the two genes is. How far apart are they located on the actual chromosome? And the way we do this is we use what's called a test cross. So you have to have one organism that is heterozygous for both the genes that you're trying to figure out the genetic distance between them. And the other parent, the other organism in the cross will be completely recessive for both the genes. So you can see in the panel on the right side, we would often write it this way: big M, big D, cross, little m, little d. So that's the nomenclature we often use. On the on the left side, they actually lay it out. They lay the alleles out as if they're on those chromosomes. And the alleles could be in different orientations. In this example, they have the big M allele on the same chromosome with the big D allele, and the little m allele on the same chromosome as the little d. But remember, the rule is just that one of the individuals is double heterozygous. So it could also be, you could also use a plant that is little m with a big D on one of the chromosomes and big M with little d on the homologous chromosome. But in this example, that's not what they started with. So they started with this parent that they show, and so we'll consider that. So when you start with this parent, most of the gametes are going to come out of meiosis having a big M with a big D and a little m with a little d. You can get something else, though, but only if there's a crossover in prophase 1 of meiosis. You'll recall that prophase 1 of meiosis is where you can get a crossover. If these genes are very close, that's not going to happen all that much. So mostly you're going to get what we call the parental allele combinations. In this example, the way the textbook wrote it, it would be big M with big D for a lot of the gametes and little m with little d. We know what the gamete types are from this other parent because they're homozygous recessive, so all the gametes are recessive. So when sexual reproduction happens, some of the gametes with a big M and big D will be fertilized by the gamete that's little m, little d, and we'll get this outcome. And then some of the gametes that are little m, little d will be fertilized by little m, little d. But what this picture doesn't show is those very few gametes from the double heterozygous parent that had that crossover in them. So we'll go to the next slide. We'll get to see that. Oops. All right. So when there's a crossover, when the two chromosomes line up, this is down here at the bottom of this figure, if there's a crossover, then you will at least get some gametes that have the crossover chromosomes. And remember, by the end of meiosis, these are monads. So you will get some. In this example, they're using A and B, but you will get some, assuming they're showing you that your parental combinations are big A with big B and little a with little b. You will get some that are big A with little b and little a with big B, but those are the chromosomes that I said are very, very few that will show up in the gametes. So the rule is that crossing over creates the recombinant allele combinations or crossover allele combinations when genes are linked. But parental allele combinations are always going to be more common than crossover allele combinations among the gametes. So this figure really combines all of it together, what a test cross really looks like. In this case, they're starting with this parent that's big M and big D on one chromosome, little m and little d on the other chromosome, and it's always crossed to a completely recessive parent. And then they're working out all the different gamete types we could get from the double heterozygous parent. There's only one gamete type from the completely recessive parent. And so when fertilization happens, most of your offspring will have big M with big D and little m little d in a genotype, or little m little d and little m little d in a genotype. But you will get a couple that result from these gametes, the crossover gametes. So you will get a few offspring, not very many, that have a big M with a little d on one of their chromosomes, or a little m with a big D on one of their chromosomes. So whatever combinations are the most common in the offspring, that really tells you what the allele combinations were in the heterozygous parent. Because the rule is parental allele combinations will always be the most common. So this is what your offspring would look like from this cross. You wouldn't be able to see the chromosomes. You would have something that's normal leaves tall, mottled leaves dwarf, normal leaves dwarf, and mottled leaves tall. And what you'll notice here is that this bottom chromosome is the same every time, because remember, that came from that completely recessive parent. But what we really want to focus on is this top chromosome, because that's really the one that's really dictating what the phenotype is. If the plant got a big M from that parent, then they have the normal leaves. If they got a big D from that parent, they're tall, because this chromosome here from the recessive parent doesn't contribute any change to the phenotype. So we focus on the chromosome that came from the heterozygous parent. And so you can see in the data, they didn't count very many here, but when you had, 100 and some odd offspring, 123 offspring, most of the offspring are in these groups, so 55 and 50 as raw data counts, and a few offspring in these other groups. So based on that, you know that the allele combinations that are most common are going to be the normal leaves allele with tall and the mottled leaves allele with dwarf. Those are parental combinations. And what the crossover combinations are the more rare ones. Normal leaves with dwarf and mottled leaves with tall. And we sort of ignore what's on the bottom here. Of course, we know they're diploid, but we focus on the chromosome that came from that heterozygous parent. So the rule is for any pair of linked genes, like M and D, or A and B, if you do a test cross between a double heterozygous parent and a completely recessive parent, from the double heterozygous parent, the gametes, the parental gametes, will always be more numerous than the recombinant gametes. And I defined parental gametes really means gametes with parental allele combinations. And so what that means is in the offspring, much larger percentage of offspring will have the combination of traits that represents those parental chromosome allele combinations.